Okay. Um, eh, Here we have the presentation of Dr. Emil Kakis from USA, Clinical Trials Overview and Evolution. It's a pleasure for me to introduce Dr. Kakis. Dr. Emil Kakis is CEO of Ultragenyx. Is best known for his work developing novel treatments for rare diseases and working on policy issues affecting rare disease treatment development through the Every Life Foundation. Emil is currently Chief Executive Officer and founder of Ultragenyx Pharmaceutical, a company with 4 approved therapies for rare diseases and 20 different diseases treatment in development. Dr. Kakis had received numerous awards, including the Roscoe O'Brady Award for Innovation from the World Symposium and the Harry Terman Visionary Leader Award from Batllo, the Biotechnology Industry Organization. Okay, Dr. Kakis, when you would like, you can start.
Very good, Jordi. And a big thank you to the International MPS family for having me tonight and today, wherever you are in the world, to talk about clinical trials overview and evolution. It's always a pleasure to come back to our long-term home in the international MPS community. Of course, the clinical trials overview could go on for many hours because there are so many trials ongoing, and I won't be able to cover all of them. So I've selected some to talk about, and particularly to focus on what I consider the really important problem, the remaining challenges in treating MPS diseases, particularly Sanfilippo syndrome. Now, one of the biggest challenges that many of you, even though you're not maybe conducting clinical trials yourself among the families, is to understand how hard it is to do clinical development when all of the children with these diseases are so different from each other. In this picture shows me with 9 of my first 10 patients in a clinical trial we conducted in 1998. And what you can see is all of these children have MPS I. They all have slightly different amounts of remaining enzyme. And you can see the clinical condition is as varied as a child on my lap who has more severe Hurler syndrome and a woman behind me who is actually a mild patient. They both have the same problem, and a variety of patients in between these two extremes. Conducting clinical trials when the patients are very different becomes challenging, and it is a source of very great difficulty in conducting clinical development. If you overview now what is happening among MPS, as you know, there are 5 approved therapies. And the thing you know is that there are some treated with an enzyme replacement therapy approved, but others not. And you might wonder, why is this? Well, the main thing that is going on right now is that all the MPS diseases that have something you can measure on the body, how a body functions, like the lung or breathing or walking or other aspects of their physical body, are the ones that can prove the efficacy and the reversal of storage more easily. But the MPS diseases that are brain focused, like Sanfilippo, and have a dominant brain phenotype, including some other ones where there is significant brain disease, is more challenging because there's less physical disease, particularly in Sanfilippo, to be able to drive approval. And because brain biology, how your brain works, is more difficult, more variable, and many times irreversible. Therefore, conducting studies is very difficult. So treating the brain and proving how it works is quite challenging. In fact, the way the rules are will make it very difficult for new treatments to succeed in clinical development. And I think today we're seeing that challenge. And I think it's very important for you to understand that, because there's something you can do about changing how we approach these treatment for brain-focused MPS diseases. So I'll touch on first, how did other MPS enzyme replacement therapies get approved? What is the burden of effort in study designs? And we'll talk about novel study designs that we've used in the MPS 7 program. And finally, we'll talk about what do we have to do to get Sanfilippo treatments developed. There's a lot of work going on, but a lot of challenges to finishing and getting the treatment that's actually available for patients. So for history here, a long time ago, in 1997, I conducted the first trial in hyaluronidase in 10 patients. That was the very first MPS trial with a novel therapy in a formal trial design. We thought at that time, based on our discussions in the US with the FDA, that a treatment trial of just 10 patients could get us approved. We then were told, no, we had to do a randomized controlled trial, which was started in 2001. And after that, there was galsulfase, idursulfase, allosulfase, that all did randomized trials ultimately to get approval. And most recently, Vistarides alpha, the last one, which was the one we did with MPS 7. If you look at each of these trials, what you'll see, something is happening that's quite important to understand. The first trial we proposed, which was at the time accepted as sufficient to get approval, was a 10-patient trial. And it was designed like the Ceridase trial for Gaucher disease, which was 12 patients. We thought this would be enough, that we'd take kids that were sick, we would measure them carefully, treat them, show their changes, particularly in biomarker endpoints, to showing the storage that had built up was being reversed, and we could get approval. But the rules changed, and now there's a demand for clinical endpoints. But clinical endpoints mean you have to deal with the variability, how much differences there are between patients. This resulted in a requirement for a 45-patient trial that just barely made it and did get approval for hyaluronidase. Subsequently, an MPS 6 trial was done using walking, the smallest trial to succeed, 39 patients. It was successful, but finding 39 MPS 6 patients was an international effort and was challenging to do. Subsequently, the trials got even bigger, 96 patients, 176 patients, in some cases longer, more challenging. And in addition to these trials, other supportive work had to be done. What you can see over these years is the cost has been going up. Now, the trial for MPS 7 was smaller, and I'll touch on a little bit why it was smaller and what we were able to do. But I think the key thing in looking at all this work is that biomarkers were not accepted for approval in any of them. So the question you might ask, well, how is it that if you look at the number of patients in these trials over time, that we went from 10 patients being enough to then needing 176 patients to get approval? And now MPS 7, we brought it back down to 12. Well, the truth was, if MPS 7 needed 176 patients, there are not that many in the world that I'm aware of at this point. It would be totally impossible. We had to find a way, and that pushed us into trying to develop another way of looking at trials and design to try to improve the efficiency of those trials. Now, many of you have heard about and talked about randomized, double-blind, placebo-controlled trials. And for many parents, particularly Sanfilippo parents, this is a horrible thing to think about, that you're going to put some kids on placebo, a saline, and show that they decline while the other patients get better. There's something that seems fundamentally unethical about this, but I guarantee you that the EMA and the FDA are demanding and requiring this. How do we do that in a way that doesn't harm patients? It is challenging. The crossover design, where you have a patient without treatment, you're monitoring them, and then you cross them onto treatment and compare before and after, can prove efficacy in my mind, but many authorities just don't believe that is a rigorous enough design. But it is a more sensitive design when the patients are very variable. To try to improve a control design, we came up with what we call the blind start design, which has elements of placebo, but is really a crossover design as well. So I want to show you that briefly as the way trial designs might evolve. In this case is a schematic of the blind start design. And basically what we do, instead of in a randomized trial, you would take one group of patients and divide them into two and then treat this half, but not this half. What we're doing in this case is dividing the patients into 4 groups. The first group, without any knowledge what they're getting, they get on treatment right from the beginning. The other 3 groups get placebo. And at times that for which the patients don't know, each group will cross over onto drug. Now the analysis is your before in red there versus after therapy for all the patients. So it's like a traditional open label trial, but since the patients don't know when they started treatment or when their assessment is, it allows us to get objective measures of what's happening because they don't know when they're supposed to start getting better. We believe this should give us an objective view. Now, here's the most important thing to understand is that this design, when you compare it to a randomized control trial, is actually 5-fold more powerful at detecting change because of the design. If you have differences between the groups in a randomized control trial, it can ruin the study. And in small trials, that happens a lot. But in this trial, all the patients get treated. They're all compared to their baseline, and therefore, you don't have that problem with some patients being who are sicker or less sick being in one group or another. All of them get treated. We think the modeling that says this is a way that we could be looking at treatment, and it's a way to gain more power. So that's one thing we approached. Now, another thing is how do you measure disease when it's variable, when all the patients are different? It's another part of the story, not just the design. And what we've been promoting lately is an alternative way of looking at measuring disease in MPS patients. Now, you know your child, and you know when you look at a child with MPS, you know they have multiple different things wrong, right? When you look at your child, you know there's a whole series of different problems. They may have lung infections, they could have hearing problems, they could have their liver's very big, they could have bone problems. You know there's multiple things. But yet our clinical trial system picks one thing because of statistics have now controlled the process to the point that we're actually forgetting about the whole patient except the one thing as a primary endpoint. And we think for a disease like MPS, that's fundamentally wrong. There are many different things to look at, and some patients have some problems and other patients have other problems. Yet you want to select people that only have one type of problem and measure those and do a trial. That's what's happening currently. In the MDRI or multi-domain responder X, it's a method that can actually eliminate that problem. In this case, you choose 4 to 6 parts or problems, clinical domains. You enroll all patients in the trial, and you score patients on these multiple domains. Are they better or worse? You're looking for big changes that improve above or below a minimum important difference. If we count only the big wins and losses in these changes, you actually will eliminate any small change for patients that don't have something or things or situations where they won't improve, and look for the big movement. And that will allow you to actually include patients that don't have every problem in the trial. And with multiple domains assessed, you capture more of what's good that's happening as well as what's bad. And that gives you more power. The more information, the better power. So I'll show you an example of that here, how it was done in a analysis of loranodida's phase 3 trial, which was presented post hoc, meaning after the trial, this analysis was demonstrated. And I think it gives you an idea how looking at multiple domains will allow you to get a better picture for how your child is doing and a better picture for how regulators think the treatment is working. In this case, we use the analysis of forced vital capacity, it's lung capacity, walking distance, sleep apnea, which is how many times you stop breathing when you're sleeping, shoulder flexion, how much you can move your shoulder, and visual acuity, how well your eyes work. And we set thresholds for what's an important difference, what improvement would be clinically important to the patient. And we only count those that go up by that much or down by that much. And we're going to look at all 5 domains. And we did not select for these in all the patients. We only selected for forced vital capacity. All the other ones had variable presence in these patients. So if you look at the trial patients here, if you look in the vertical columns at the top, this is the lungs, the walking, shoulder, sleep apnea, and visual acuity. On the left are the 23 patients who got placebo, and on the right is the hyaluronidase treated group. What's important here is to know when there's an improvement, it's red. When there's a decline, it got worse, it's a yellow. And there's no change, it's really gray, it's supposed to be. And the blue, light blue, is just, there's no data available for whatever reason. If you look at this, you and I, all of us, can understand the data right here looking at this heat map. On the left, on placebo, you can see there's as much yellow of decline, maybe more yellow than red, which tells you among these different domains, patients are getting more, on average, worse. On the right, you can see many more domains that are red than yellow. That means that the majority of the changes are actually improvements in the Aldurazyme group. And you can also see which improvements were seen in each patient. Like some patients you can see had 3 domains improved, which is a pretty important result, but some didn't. Some had only 1 domain and got worse, so there is some variation in who responded. But the value of this is that if you look at the co-primary endpoints of the trial, there were 13 net domains positive. By looking at 3 other domains, we added another 9 domains, right? That means 69% more power, more information to determine what's happening. And I think if you look at this picture, you have a better idea what's happening in these patients than you do from almost any other type of analysis. Now, we took the trial design I talked about, the blind start and the MDRI, and we put both into the MPS 7 trial. The MPS 7 is extremely rare, and we had to do our best to be able to do a design that would allow us to detect efficacy. When we did the trial, we treated these 12 patients in the blind start design, and we looked at their biomarker, and this is the urinary GAG, or the MPS-like secretion in the urine, and we showed a very nice reduction, as you can see shown here, 65%, very small error bars, meaning everyone responded very similarly, even though the patients were age 5 to adulthood, very wide range. Half of them didn't walk, they were already in wheelchairs, half could walk. So a very variable population, but they all have a good biochemical effect. When we look at the MDRI shown here in the box, out of the 6 patients that could walk, there were 3 that had 60 to 80 meter improvement in walking. So that was considered a clinically meaningful improvement. And we added up other things like the small motor fingers function and other aspects of gross motor function in addition. And if you add up all of these blue domains, we ended up with basically 9 domains positive, 3 domains negative, or a ratio of 3 improvements to 1 decline in the trial. If you added fatigue to the score, it even added another 4 domains. What was important when we looked at this is that with this analysis, with a 12-patient study, we actually showed a statistically significant result by adding up these improvements across many different domains. I think that is a miraculous result to be able to show improvement with a 12-patient trial. And this is a disease where these are the 12 we could find who would enroll. So with nothing, we couldn't do any more in that. So the MDRI's advantages is that you can study in unselected variable populations, more mild and severe. You look at multiple domains, look at more things. And if some of your patients have a problem or don't have a problem, it doesn't affect the analysis. So you don't have to have all the patients have all the medical problems, which rarely happens. And it allows you then to include all comers. Everyone can be it. It also means less guesswork on the endpoint choice. What if you pick one and it's not the best one, it turns out? Well, in this case, it doesn't matter. You pick 5. If 2 of the endpoints become non-assessed, it doesn't matter. The other 3 can score and you can end up with a success. So it's more robust against accidental variation that happens a lot when you do clinical trials, because you don't know who's going to be in the trial until you finally get there. Now, I want to talk about one other issue. In the early days of the MPS work, we thought biomarker approvals were going to be important. that we can measure lysosomal storage or the excretion of MPS or glycosaminoglycans and prove the biochemical efficacy of the drug. And that was really the underpinning of treating MPS diseases. And because there's so much variation, the biomarker-based approval would be far more powerful at figuring out the problem. What is difficult in Sanfilippo is that many patients, by the time they're diagnosed, have substantial irreversible disease. Now, if you want to treat them earlier, that would work, but if you could have newborn screening. But no one wants to do newborn screening until you've proven a treatment. So if you can't prove the treatment until you diagnose early enough and you're not doing the screening to find them early enough, then you've created a situation where it's basically impossible to get forward. And so to do this means finding those few patients diagnosed early and following them for many years until they decline or not. And the challenge is that when you're doing that, no one wants to be in a control group. And so people want to use natural history controls, right? Historical control. But the truth is, for slowly declining disease, that doesn't work very well. So we have to be better at this. We have to figure out how to use our biomarkers and other things to help get approval. Now, I'm going to skip ahead and talk a little bit about biomarkers, but I want to focus here briefly on the fact that no matter how severe you are, rapidly, intermediate, or slowly, you can look at the biomarkers at each point in time, that's the green bar, and show a good improvement. But what the clinical changes at each stage of a patient's life can be highly variable. They could be very irreversible, or it could be they don't have very much disease yet, and therefore hard to measure. We have to understand that the clinical variation creates a problem, and particularly one for Sanfilippo. And I want to show you on the next slide in Sanfilippo. Now, this looks technically complicated, but imagine your child's up on the upper left here, and you see that there's a starting point, their intellectual function's good. And early on, before you even diagnose them, if you treated them there, you could maintain them, but you wouldn't really show improvement because they hadn't declined. It's very hard to demonstrate what's happening, and it may take many years to show that they're going to decline. Now, what normally happens, you're farther down this curve and you start treating there, but at that point in time, it could be that half of the decline they've already had is irreversible decline, and the improvement is relatively small and variable amount. Doing a trial then is very difficult here. It's more only about demonstrating stabilization than it is about improvement. But then showing stabilization compared to control becomes difficult to do. It's this problem with the clinical endpoints that make it very difficult to do Sanfilippo. And if you look to trials going on today, and there's a number of them, there was the Shire-Takeda intrathecal idursulfase study, which failed, looking at the differential ability scale and some other clinical endpoints. That was a pretty good sized trial. And that's very challenging with the drug, where I know that the quantity of enzyme being given is going to clear the brain storage. But that variation is because the brains already have injury. Some can improve, some cannot. And that variation makes it hard to do a trial. There's other trials going on, another enzyme from Denali. There's gene therapy work, such as from Abeona or lysogene. All of them are looking at improvements in heparan sulfate to varying degrees, but proving, good efficacy is tough. I think Abeona has published or presented 3 young kids that were doing well and retained cognition over 3.5 to 5 years. But that's going to be very hard to get approval. So how are we going to get there? The truth is, if we don't start accepting biomarker approvals, it'll be very hard to get approval in Sanfilippo for any of these diseases or any of the CNS. It is a big challenge. So we need to start the walk toward medical evolution. What that means is we need to start accepting the use of biomarkers and their imperfection to get started and start treating, and then to get newborn screening and treat earlier. We need to start this process. We cannot expect perfection in these treatments, and we need to get regulators to understand this. If we do expect perfection in large randomized trials and clinical endpoints, we will have a very difficult time seeing Sanfilippo treatments developed. And I think right now there's a risk that they won't get developed as companies attempt to make progress and find themselves in trouble. We need to approve treatments when we get good heparan sulfate reductions using very specialized measurements of heparan sulfate that are highly controlled and very precise. That means good science. And we need to then look at long-term sponsored follow-up studies in the post-marketing to show what the clinical benefit is over time. If we start treating babies earlier, diagnosed earlier in life, we can hopefully change the next generation. I ask the whole MPS community needs to band together with the other neurologic genetic disease who are in the same difficult situation, like Niemann-Pick C and ceroid lipofuscinoses. You need to get together and start talking to regulators about accelerated approval or conditional approval based on biomarkers if we want to see treatments developed. That's all for today, and thank you for listening.
Thank you, Dr. Kakis. Congratulations for your presentation. It's fantastic. And I don't know if I have question for you. First of all, I would like to comment that MPS 3, for example, Sanfilippo disease, is very difficult. It's very difficult, and that you comment before, it's very important the screening before and early diagnosis, no? Because Sanfilippo disease, for example, and Hunter and another syndrome, that do you think it's very positive to have a very good treatment and to work very well in the patient?
Oh, I think it's very possible to treat Sanfilippo disease. I just think we have to understand our expectations have to be aligned with the biology, that if the kids are being treated when they're 6 or 10 years old, that a lot of neurons have been lost or are so damaged they can't recover. And therefore, you can't expect them to get better. But here's the problem. One 6-year-old is not like another 6-year-old or another 6-year-old. Therefore, you'll see variable data. And what happens is the regulator says, see, the biomarker is the same in all, but the clinical outcome is different. Therefore, the biomarker is wrong, but they are wrong. The problem is not that the biomarker is wrong. When biomarker is correct, the brain was treated. It's just the outcome that will be variable because the clinical condition of the patient will have changes that cannot be improved. We need to get our heads around this. We also need families to understand this, that we can't get to the end of this in Sanfilippo without accepting the biomarker approvals and driving the best science we can and getting treatment started earlier.
But it's curious, but in the pandemic with COVID-19, all is very fast, but it's very difficult to have a new treatment if MPS disorder, for example. What is your opinion about this?
Jordi, I didn't quite understand the question. Please say it again.
But for develop a new treatment, if MPS is very difficult to have a new opportunities. But in this pandemic with COVID-19, okay, coronavirus, is very easy to have all the companies develop a new opportunities of treatment.
Yes, well, there are several reasons for COVID. The onset of an infection and your measurement of it is very easy, very fast, and more consistent among a lot of people. And that makes it possible to run a 40,000 vaccination study in a few months. The truth is, Sanfilippo is it takes so much time, so much people, so many sites, so much that it's very much harder when you have a rare disease. If we had a marker like you do for infectious disease that they would accept, I think that would greatly improve the prospect of getting treatment. I would say though, Jordi, we are at an exciting time. There are many patients, many trials that have been underway, but I'm warning everyone there is a risk that these trials may fail to achieve enough efficacy on the traditional terms of efficacy to get approval. And that will be a disaster that could come and harm all the patients and families hoping for a treatment.
Okay. And I have a question for one family and say that if you have a very opportunity to treat the syndrome, it's very easy to have the diagnosis before or to offer treatment, or the doctor don't have to have a result very easy for the treatment?
Well, I think the main challenge for Sanfilippo is that it's actually recognizing the clinical symptoms and making the diagnosis early, because the symptoms can be very nonspecific. And so the problem is, by the time they are diagnosed, their clinical problems may be substantial, and the ability to reverse them short. They may have a relatively short window. If you remember, in the early days of MPS I bone marrow transplant, if you remember the early days, they were transplanting older, younger patients at different ages. And very quickly they realized they had to do transplants before age 2. If you remember this whole story, that the standard was set, you have to be before age 2 for safety, but also because it wasn't working in the brain if you did it after that. You couldn't save the brain. The same thing exists in Sanfilippo. There is an age where you start losing ground. Whether it's a precise age that is the same for all patients, I don't think that's true, but there is a window of time that gets lost. And I think this is what's going to make it so hard to try to enroll a study of a particular age and not to get stuck with variable data like the Hunter CNS treatment, which was a good sized study, and still fail. I think this is the challenge we all have. We have a lot of trials going on, but how are we going to get to the end?
And last question. Do you think that the regulators are receptive to change their criteria? What can we do as patient organization to change this criteria within our communities?
Well, I think within the European community, within the US community, the groups with MPS, Sanfilippo, CNS problems, brain problems, and other brain disease need to start lobbying the regulators directly. They need to request meetings, they need to talk to them and warn them about the pending disaster that all these treatments, which are scientifically sound, delivering the missing enzyme, will fail if they do not properly understand the science and start approving products based on science. I recently wrote an article in Stat News, and I praised the Alzheimer's approval, which was based on biomarkers. And I said in the beginning, treating something like Alzheimer's is very hard. The decision seems imperfect, and people are unhappy. But the truth is, in Sanfilippo and other diseases like it, we need to start making those approvals and driving things forward. Otherwise, we'll not do the long walk to medical evolution and end up with a great treatment and a diagnosis before age 1 and a great outcome for the next generation of Sanfilippo kids.
Thank you, Dr. Kakis. Thank you for to share the information with us. It's impressive. Okay, we have another table. Okay.
Thank you very much.
Thank you, Emil.